Now that we have introduced the general stages of development and some of the history of developmental biology, we're going to take a closer look at the factors that are important for regulating the early stages of development. And we'll begin by discussing the maternal control of cleavage, because what do you know? For some organisms, mothers have a say even on how the organism divides as an embryo. What I mean by this is that in most species, there are mRNAs that are of maternal origin and are present in the egg before fertilization, and these mRNAs control the initial rate of cell division and the location of the blastomeres. In many organisms, these initial cell divisions, which are controlled by the maternal factors, happen very fast. And as control of cell division switches over to the embryo's genome, then cell division rates slow down. That point at which development control is switched from the maternal to the embryonic genome is known as the Midblastula Transition, or MBT. During the early cell divisions, when the rate of cleavage is controlled by maternal factors, the volume of the cytoplasm doesn't increase. Instead, the cytoplasm of the zygote is divided into smaller cells. This means that at first, the nuclear to cytoplasmic ratio is low, since the volume of each cell is very large. As cell division progresses, the nuclear to cytoplasmic ratio increases as that cytoplasm is divided into more and more cells. Recall that this is related to the fact that in certain embryos, the G1 and G2 phases of the cell cycle are skipped. Now, this fast rate of cleavage is thought to be an adaptation to generate a large number of cells very quickly. Another aspect of cleavage that's controlled by maternal factors is the direction of cleavage, which is dependent on the planes of cell division and the orientation of the blastomeres relative to each other. But these patterns of cleavage are not only controlled by maternal factors, but also by the presence and the distribution of yolk. And the terms used to describe these patterns of cleavage were actually coined by Aristotle. These terms are holoblastic and neuroblastic. Organisms whose eggs have little yolk undergo holoblastic cleavage. So this means that the entire fertilized egg undergoes cell division. The prefix holo comes from the Greek word complete. So holoblastic cleavage simply means complete cleavage. On the other hand, organisms whose eggs have a lot of yolk undergo meroblastic cleavage, which means that only part of the fertilized egg, the part that has less yolk, undergoes cell division and contributes to the developing embryo. Now, the prefix mero also comes from the Greek, and it means partial. So meroblastic cleavage is partial or incomplete cleavage. The reason why cleavage is complete in embryos that have little yolk and incomplete in embryos that have a lot of yolk is that yolk inhibits cell division. As we'll see in a moment, there are eggs that have sparse and even distribution of yolk, and eggs that have uneven yolk distribution. And these are named, as we'll see in a moment, based on the amount and the location of that yolk. In eggs that have an uneven yolk distribution, the side that has less yolk is known as the animal pole, and the side that has more yolk is known as the vegetal pole. The presence of yolk in the vegetal pole inhibits membrane formation and cell division. In addition to that, the nucleus is actually displaced to the animal pole, so this is where cell division will commence once the egg is fertilized. Recall that depending on the amount of yolk that's present in an egg, cleavage will either be holoblastic or meroblastic. Holoblastic cleavage, in which the cleavage plane extends through the entire egg, is seen in isolecithal eggs, and these are eggs that have a sparse and even distribution of yolk. To help you remember this term, keep in mind that the prefix iso means equal. As you can see, isolecithal eggs can undergo different types of cleavage. For example, mammals have isolecithal eggs which undergo a type of holoblastic cleavage that's known as rotational, while annelids or segmented worms also have isolecithal eggs, but they have a different cleavage pattern that's known as spiral. Because these embryos have little yolk, they need to depend on another nutrient source for feeding. So they either generate a voracious larva, or in the case of mammals, they rely on placental feeding. Something else that is apparent from this diagram is that holoblastic cleavage is also observed in fertilized eggs that have moderate amounts of yolk and an uneven distribution of that yolk. And these eggs are known as mesolecithal, with the prefix meso meaning intermediate, since these eggs have an intermediate or moderate amount of yolk. This type of egg is observed in amphibians. And as was alluded in the previous question, cell division in these eggs happens slightly faster in the animal pole as opposed to the vegetal pole because there's more yolk in the vegetal pole. By the way, you don't need to memorize these sub-classifications like radial, spiral, bilateral, rotational. What you should know is that holoblastic cleavage is observed both in isolecithal eggs, which have a sparse and even distribution of yolk, and in mesolecithal eggs, which have moderate amounts of yolk that is unevenly distributed. So the difference between those is that in the mesolecithal eggs, even though the cleavage is holoblastic, it will be displaced so that it happens faster in the animal pole as opposed to the vegetal pole. Unlike the isolecithal and mesolecithal eggs, telolecithal and centrolecithal eggs have a large amount of yolk, with only portions of the egg undergoing cleavage after fertilization. This yolk serves to provide nutrition for these embryos during embryonic development. Zygotes that have a large amount of yolk undergo neuroblastic or incomplete cleavage. So cleavage does not penetrate the yolky portion of the cytoplasm because, remember, yolk platelets impede the formation of membranes. Organisms whose fertilized eggs have these large amounts of yolk and thus undergo neuroblastic cleavage include insects as well as birds, fish, and reptiles. In the case of birds, fish, and reptiles, as well as mollusks, their eggs are telolecithal. This means that most of the egg has a large amount of yolk, and there's only a small region of the egg that lacks yolk. This region is where cell division happens. The prefix telo means extremity. So the word telolecithal suggests that the yolk is at one end of the egg. In the case of insects, their eggs are centrolecithal. This means that yolk is located in the center of the egg. Now, you may look at this image and wonder how come there seem to be cells at the center of the egg if this is where most of the yolk is found. Well, we need to remember that cells are three-dimensional. So what is happening here is that the cleavages are superficial. The cells form a sort of jacket around a yolky center. And as we'll see later, the early cleavages in Drosophila only involve nuclear divisions without cytokinesis. Besides the effects of yolk on the pattern of cleavage, there are also inherited patterns of cell division. This is why zygotes that have similar amounts and distribution of yolk, like all of these isolecithal eggs, still undergo different patterns of cleavage. All of these fertilized eggs are undergoing holoblastic cleavage. The cleavage furrow extends throughout the entire zygote. However, the organization of the cells or the angle of the cleavage is different. The next phase of early development is gastrulation, and we'll be looking at the patterns of gastrulation of different organisms throughout the semester. And we'll see that gastrulation involves a series of combinations of different types of cell movements. These figures, for example, show the gastrulation of the amphibian embryo. And as you can see, the mesoderm, which is shown in red, and the endoderm, which is shown in yellow, those two are being surrounded by the ectoderm, which is shown in blue. And gastrulation is an animal adaptation. It's not observed in plants or fungi. So the cell movements that we'll discuss apply only to animals. Although patterns of gastrulation vary among animal species, all patterns are a combination of these 5 basic types of cell movements. In invagination, cells enter into the internal cavity of the blastula, which is known as the blastocele, and this movement pattern resembles a ball being poked. Involution involves the infolding of an external sheet of cells so that these cells end up spreading over the interior surface of the cells that remained in the exterior. Ingression, on the other hand, refers to the movement of cells into the internal cavity of the blastula, but the cells are moving as individual cells. These cells are usually attached to each other at first, so they have to lose their adhesion for each other in order to move into the embryo as individual cells. The lamination is similar to ingression, but it involves the movement of a sheet of cells rather than individual cells. So you go from one layer of cells to 2 layers or multiple layers. Finally, epiboly refers to the spreading of an outer sheet of cells, which are usually ectodermal cells, to cover more interior cells. So epiboly involves the division of cells as well as changes in how these cells associate with each other, so changes in what is known as intercalation, as we'll see later in the semester. It's important to remember that combinations of several of these movements happen simultaneously over the entire embryo, so these different movements need to be coordinated. As I mentioned previously, gastrulation results in the formation and the segregation of the germ layers, the ectoderm, the mesoderm, and the endoderm. The ectoderm gives rise to the epidermis of the skin, which is the upper layer of the skin, and it also gives rise to the brain and the nervous system, as well as a population of cells known as the neural crest, which gives rise to many different tissue types, and we'll talk about those later in the semester. The mesoderm gives rise to the kidneys, muscle, bone, the circulatory system, as well as the connective tissue and the gonads. Now, I want to be clear that the gonads are the testes and the ovaries. They're not the same thing as the germ cells. The germ cells, the sperm and the egg, are not considered part of any germ layer. I should also point out that when I say circulatory system, I mean not only the heart and blood vessels, but also the blood itself. Finally, the endoderm gives rise to the lungs and the digestive system. Although we consider the germ layers as distinct from each other, the development of the structures derived from these germ layers doesn't happen in a vacuum, and it involves interactions among cells of different layers, as we'll see throughout the semester. Gastrulation also accomplishes the establishment of the 3 major axes of the body: the anterior-posterior axis, or head to tail or mouth to anus axis, the dorsal ventral axis, which refers to the back and belly, and the right-left axis. This last axis is significant, though we may not really think about it very much, but we're not completely symmetrical. The placement of organs matters, as we'll see later. Now, since the movements of gastrulation are difficult to visualize just by looking at pictures, take a look at the following videos to get a better sense of what this looks like in the embryo.